This video is going to recap single gene mutations and also chromosome structure mutations. So we should remember what mutations are from Nat 5 biology. These are random changes to the genetic material. And single gene mutations are the first one that we're going to talk about. And these are mutations that affect a single gene. And this means that only the protein that that particular gene codes for is going to be affected. And we need to be able to describe what effect these single gene mutations have on the gene itself or the DNA, and then what impact that would have on the protein that is being produced. So what impact did the change in DNA have on the sequence of amino acids at the ribosome? And if we look at our starting sequence of DNA, we can see that we have these triplets called codons, and each of these are going to code for a different amino acid at the ribosome. And we need to be able to describe what effect a mutation has on that sequence of amino acids. Okay, so the first type of mutation that we're going to talk about is a substitution mutation. And remember, we have to think about what effect these are going to have on the gene and what effect that they are going to have on the protein. So a substitution mutation is when one nucleotide is swapped for another, and therefore one codon is different. So I'll show you what that looks like. Remember, these were our codons, and these were going to code for specific amino acids. In our starting sequence, we have a G here, but in our substitution mutation, that G has been swapped for an A. So what that means is this codon is now going to code for a different amino acid. So our protein is going to have one different amino acid, and all the other amino acids are going to be the same. We call this a missense mutation. So that's our first type. The other type that can occur is we can have a substitution mutation where we change one nucleotide, and what that means is we now code for an early stop codon. So one nucleotide swap for another, and an early stop codon is produced. What that now means is we are only able to make the amino acids for this little section of our gene here because of that early stop codon, and therefore our protein is going to be shorter. And we call that a nonsense mutation. So the substitution mutations are missense and nonsense. So the next two we're going to talk about are insertion and deletion, and collectively we're calling these frameshift mutations. And these are going to have a bigger impact on our protein. So again, we'll look at what this looks like. So again, we've got our codons marked out here, and remember they're going to code for a specific amino acid. Now in frameshift mutations, we are either going to add or we are going to remove a nucleotide. So our starting codon was AGC here, and in our deletion mutation we have removed that G nucleotide. Now what that means is everything is going to shift back one. So instead of coding for AGC, we're now coding for ACG. Instead of coding for GCT, we're now coding for CTA. And it's going to be the same for every single amino acid after that point of mutation. They are all going to be different. So bigger impact on our protein there. And for our insertion mutation, again, we started with AGC, but we have now added this T nucleotide in here. So again, instead of coding for AGC, we're now coding for ATG. And then instead of GCT, we're now CGC. Everything has shifted along one. So again, what that means is every single amino acid after that point of mutation is going to be different and bigger impact on our protein. So another type of substitution mutation that we need to know about are splice site mutations. And remember, substitution mutations are when one nucleotide is swapped for another. So we know from our gene expression topic that when our cells make proteins, they copy the DNA into mRNA. And the mRNA had these extra non-coding regions called introns that we cut out in that process of splicing. And we just put together our exons, our coding regions, so that we could make our protein. So our cell knows where to cut out these introns using splice sites. So that's just little sections of our mRNA that tell us where to cut out our introns. And sometimes mutations, substitution mutations, can occur at these splice sites, which mean those signals go wrong. So it ends up that we actually retain this intron in our mature transcript. And what that would mean is, again, we've got those extra little non-coding regions that are now going to be coding for these amino acids. So we would end up with extra amino acids here. And sometimes a splice site mutation can occur, which means that we actually remove exons as well. So if we removed an exon, it would mean that we had less amino acids in our final protein. So again, all of these would lead to a faulty protein. So just a recap about SQA wording before we attempt a question. You could be asked about the effect of a single gene mutation on the gene, in which case we are referring to the nucleotides and codons. And you could be asked about the effect on the protein. You're referring to the amino acids or the length. So remember our three types. We had missense, we had nonsense, and we had splice site mutations. So if we were asked about the effect of a missense mutation on the gene, again, we're referring to our nucleotide. So one nucleotide was swapped for another, and therefore one codon is going to be different. Effect on the protein, because we've changed one codon, one amino acid is going to be different. For our nonsense mutation, again, it's substitution. So one nucleotide is swapped for another, but we produce an early stop codon. And because we're producing an early stop codon, our protein is going to be shorter in length. Splice site mutations were a substitution mutation that occurred at a splice site. So remember, splice sites were those little instructions to cut out the introns. If we retained some introns, it would mean that the effect on our protein was that we had extra amino acids. And if we were to cut out the exons, that would result in some exons not being included. We would have less amino acids. So thinking about frameshift now, if we are asked about the effect on the gene, again referring to nucleotides or codons. And for the protein, we are thinking about amino acids or length. And we had two types. We had our insertion mutations and we had our deletion mutation. So if we are asked about the effect of a frameshift mutation on the gene, for insertion, one nucleotide has been added, and therefore every codon after that point of mutation is going to be different. Effect on the protein, every amino acid after that point of mutation is going to be different. For deletion mutation, one nucleotide has been removed. Every codon after that point of mutation is going to be different. Everything shifts along. And for our protein, every amino acid is going to be different after that point of mutation. Okay, so this is an example of an SQA question. You can pause it if you want to read through it properly. But we're being told that we've got this particular disease in humans and it is caused by this frameshift mutation. So immediately I'd be highlighting the keywords and thinking that I'm either talking about an insertion mutation or a deletion mutation. And we're being asked about the effect on the lactase gene and on the structure of lactase. So sometimes the SQA wording is going to be slightly different here. You could be asked about the effect on the gene. Sometimes it will say on the mature transcript. That is exactly the same way that we would approach answering it. We're still talking about our codons and we're still talking about our nucleotides. And instead of asking about the effect on the protein itself, we've just been asked about the structure of lactase. That is the exact same thing as asking about the effect on the protein because lactase is a protein. So again, we're thinking about our amino acids for effect on the protein. So frameshift mutation, what you would say for effect on the lactase gene is that we are adding or removing a nucleotide, and therefore every codon after that point of mutation is going to be different. And for the effect of on the structure of lactase for your protein, because we've changed every codon after that point of mutation, every amino acid after the point of mutation is going to be different. So we approach all of these questions in the exact same way, effect on the gene and effect on the protein. So here we have a mutations question that revolves around an mRNA codon table. We should be familiar with that from gene expression. And we're being asked which of the following mutations in a section of mRNA would result in the production of a shortened protein. So remember, shortened protein we're thinking about our nonsense mutation, and we're looking for the presence of an early stop codon. So identify where our stop codons are in the table, and then what are the sequences? So we've got UAA, we have UAG, and we have UGA. So if we look at our original mRNA codons here, and then our mutated mRNA codons, we are looking for the presence of one of these codons here. So having a little look, we can see that in B we had UAC to start with, and in the substitution mutation we had UAG being coded for. So again, we have that early stop codon being produced. We wouldn't be able to produce the amino acid that is coded for here due to that presence of the early stop codon. So chromosomal mutations are mutations that can affect several genes. We're looking at entire chromosomes here. And we've got 4 different types that we need to know. We've got deletion, duplication, translocation, and inversion. So if we look at our original chromosome, we've got 6 different sections here, and these sections are going to be coding for lots of different proteins. They're going to contain multiple genes. So we've got A, B, C, D, E, and F. In our deletion mutation, we say that a section of a chromosome has been removed. So we can see that we no longer have this B section here. We are not going to be able to make any of the proteins that are coded for by the genes in that section, and that can have a huge impact on the functioning of our cells. For our duplication mutation, we need to know that chromosomes come in pairs. We call these homologous chromosomes. So there's going to be an exact copy of this original chromosome that we're looking at here. For our duplication mutation, we say that a section of a chromosome is added from its homologous partner. And we can see that there we have 2 B sections in our duplication chromosome there. So if we look at translocation here, we can see we've got 2 different chromosomes and they've got a little bit muddled here. So we say for translocation, this is where a section of a chromosome is added to a chromosome, not its homologous partner. So for duplication, remember that was our homologous partner, so we get 2 B sections there. For translocation, we've now got J, K, and L, and we've got G, H, and I. Those have been added from another chromosome, not its homologous partner. And then the last one to look at is inversion. So for inversion, this is where a section of a chromosome has been reversed. So we can see that here, we've completely flipped this little section. So a section of a chromosome has been reversed. So just looking at the effect that these mutations could have is going to completely depend on where our chromosomal mutation occurs. If it was to occur at the end of the gene, it could still mean that we can make all those proteins that the genes code for. Whereas if it was to occur in the middle of a gene, and let's say in translocation, we are moving from a chromosome that's not its homologous partner, if it had broken off right in the middle of a gene, we would no longer be able to code for those proteins. That gene would be interrupted. If we look at duplication, for example, we've not actually lost any information there. We're still able to produce all of the proteins that those genes code for. We'd just be producing extra. In deletion, we are producing a shorter chromosome, so we can see that we're missing that little B section there. We're missing all of the genes that are going to code for various proteins within that cell, and therefore we're losing genes and those proteins wouldn't be made. So it's going to have a massive impact overall. So we've kind of talked about mutations having these negative impacts on our ability to make proteins and causing things like disease, but they can actually be really beneficial because they provide new variation to an organism by introducing these new alleles. Thinking about our chromosome duplication mutations, we're duplicating an entire gene, and sometimes the second copy of that gene that we've produced can be altered, and produce new DNA sequences. So if we look at this example here, we have this Antarctic fish, and as a result of duplication mutations, it's developed these antifreeze proteins, which allows them to survive in these really cold environments. So again, that's really beneficial for them. If we look at their ancestral genes, they've got this gene A here, and as a result of a chromosome duplication, we've got 2 copies of this gene A. And in one of those copies, a mutation might occur that allows it to produce these antifreeze proteins, which we can see in this new gene C. So the new gene C has this mutation that allows it to produce antifreeze and allow it to survive in those really cold environments. We've still got this other copy, we've still got gene A, still got all the other genes that were present and continue to function as normal, but we've got this new gene sequence that allows them to have the selective advantage. So we say that duplication mutations are one of these key driving forces in evolution. It allows these beneficial mutations to occur in one copy of the gene, and the other genes can still be expressed to produce those original proteins. So just have a go at an SQA past paper question. We're being asked which type of chromosome structure mutations have occurred in these chromosomes. So looking at our original chromosome, we have genes 1 to 10. And if we look at chromosome P, we can see that we've got 9 and 10 twice. So we've added a section of a chromosome from a homologous partner. So we would say that that was duplication. We're between A and B. For Q, we can see that we're going 1 to 6, and then we've got 10, 9, 8, 7. So we have reversed a section of the chromosome. So that would be inversion. And then just check R, we're going 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then we have this new gene sequence here. We've got 15 and 16. So that is a section of a chromosome that has been added from a chromosome that is not its homologous partner. It would be translocation.